Okay, I think we should be live now. Can we get a confirmation from the chat? Can anyone see anything that's happening right now? Ooh, I hope so. Yes, great, wonderful. All right, guys, welcome to what I'm really excited to present as the first Neuro Launchpad session, which is going to be 1 out of 10. This is just the beginning of the season. We're super excited, just really excited, honestly. And we have 2 great talks to start us off. And really the goal of this is to give trainees exposure, to highlight trainee neuroscience from across the country. If this goes well, if the 10 weeks go well, which you can find at our website at www.neurolaunchpad.com, we hope to repeat this process and really make it something permanent that people can keep coming back to, and we can keep giving exposure to trainees, both postdocs and graduate students that are doing neuroscience across the country and hopefully the world in the future as well. So thanks a lot to Worldwide Neuro for hosting us, and we're just going to get right to it. If you have any questions, please submit them in the chat. Today's session is going to be moderated by David Schneider, who's faculty at NYU, and Jenny Lee, who is a friend and a graduate student at Duke University. And I'm just going to hand it off to David. So David, go ahead.
Great, thanks, Jenna. So welcome, everybody. It's really an honor to be the faculty host at this inaugural session of the Neuro Launchpad. When Jonah asked me to host this session, I was thinking back on it. It was just a couple of months ago that I think I got like a text message from Jonah that was like, hey, do you think there'd be any interest in a series of talks given by trainees for a broad neuroscience audience? And my gut reaction at the time was that is exactly the same as my feeling now, which was, hell yeah, there is definitely going to be an audience for this. I think it's an awesome thing. And at the time, too, I knew there was no doubt that Jonah would put together a really great group of co-organizers and that they would put together this really cool series. So kudos to them for doing it. So with that said, it's my absolute pleasure to introduce our first speaker of the day, which is Sebastian Choi, who's a postdoc in David Ginty's lab at Harvard Medical School. And in the Ginty lab, he combines behavioral, physiological, and anatomical approaches to study touch and pain in spinal circuits. So Sebastian earned bachelor's and master's degrees at the Korean Advanced Institute of Science and Technology before moving to Harvard for his PhD, where he studied molecules and mechanisms of synaptic transmission in the nematode C. elegans. So then after finishing his PhD at Harvard, he did like many people do, because Harvard is kind of this black hole that keeps people there. He stayed at Harvard for his postdoc. And he joined David Ginty's lab. And Sebastian has been awarded, since he's been a postdoc, the Association of Korean Neuroscientists Postdoctoral Award, as well as a Blavatnik Biomedical Accelerator Pilot Grant, among many other awards. Sebastian's also an active mentor and teacher, both in and outside of the lab. He's mentored technicians, undergraduates, and rotation students in the Ginty lab. He's reviewed abstracts for the annual Biomedical Research Conference for minority students. And he's volunteered his time to teach science at local schools that serve underrepresented communities. So he's really an exceptional scientist and really an exceptional person. I'm looking forward to hearing his talk. I'm going to pass the mic to him now so that he can tell us about his work looking at parallel ascending spinal pathways for affective touch and pain. Sebastian.
All right, let me share my screen. All right. So first, thank you for the generous introduction, David. And also, first, I would like to thank the organizers for giving me this opportunity to share my work today. So my name is Seungwon, and people call me Sebastian, and I'm a postdoc in David Ginty's lab at Harvard Medical School. So today, I'll talk to you about the work that I've been doing past few years, focused on the ascending spinal pathways that convey affective touch and pain signals to the brain. So every day, we experience a myriad of somatosensory stimuli, the hugs from loved ones, and a warm shower, a mosquito bite, and the sore muscles after a workout. All these tactile, thermal, and noxious signals are first detected by sensory neurons distributed throughout our body, And then these signals are propagated into the spinal cord where they are processed and then transmitted to the brain through these ascending spinal pathways. The primary sensory neuron subtypes then innervate the skin and detect a wide range of somatosensory stimuli have been identified and characterized. However, very little is known about how the somatosensory signals are integrated and processed within the spinal cord, and also how these signals are conveyed to the brain by spinal cord projection neurons to generate our somatosensory perception and the behavior responses. So the goal of my study was to better understand the circuit logic underlying these ascending spinal pathways. So among different ascending spinal pathways, I've been studying this anterolateral pathway that is known to convey mostly pain and temperature information from the periphery to multiple regions in the brain. And I particularly focused on this one brain target and the projection neuron that innervate the lateral parabrachial nucleus. And this pathway is termed spinal parabrachial pathway, or SPB pathway. So today's talk, I'll describe two main findings of my study. So first half of my talk, I will talk about these two parallel ascending circuit modules that cooperate to convey nociceptive signals to the brain. In the second half of the talk, I'll talk about this newly identified mechanosensory limb of the anterolateral pathway that conveys an affective component of touch signals. So the current view of the ascending pain pathway emphasizes the involvement of the TACR1 positive projection neurons in transmitting nociceptive signals from the spinal cord to the brain. However, the therapeutic strategies in humans that target these TACR1 neurons or the TACR1 receptor itself have been minimally successful, indicating that there may be additional TACR1 negative projection neuron populations that convey nociceptive signals from the spinal cord to the brain. So to identify this unknown and new subtypes of anterolateral pathway projection neurons, I conducted in silico screening of over 1,300 back GFP transgenic mouse lines from the GenSet project for the ones that exhibit GFP expression in the neurons located in the superficial lamina of the spinal cord dorsal horn, where these projection neurons are enriched. So I imported 5 promising GFP lines, and it turned out that one of the lines, the GPR83 GFP, labels a major subset of spinal parabrachial projection neurons. So this is how the GPR83 neurons are distributed in the dorsal horn of the spinal cord, and they are enriched in the superficial lamina of the horn. So we generated mouse genetic tools to label either TACR1 or GPR83 projection neurons. And with the tools that we generated, we were able to visualize these two populations at the same time. So here in these images, TD tomato cells represent TACR1 positive projection neurons labeled by this combination of mouse genetic strategy and retroflippovirus injection into the PVN. And GFP cells represent the GPR83 neurons. And these, as you can see here, these two neurons are largely non-overlapping populations. And then I looked at the brain target innervation in the PVN. So PVN, the lateral parabrachial nucleus, is a complex Pontian structure. And Cliff Saper and others have shown that the PVN is comprised of several cytoarchitecturally distinct subnuclei, which have distinct input-output connections with other brain regions. So I first look at the synaptic terminals originating from the old spinal cord neurons using this caudal body part specific Cre lines in combination with Cre-dependent symptophizing TD tomato mouse line. So as you can see here, most of the subnuclei within the PVN receives synaptic inputs from the spinal cord, except the VL and the central region of the EL. And then next, I directly look at the synaptic terminal distribution of either TECR1 or GPR83 SPB neurons. And it turns out that TECR1 positive SPB neurons form synaptic terminals mainly within these two subnuclei, IL and CL, whereas GPR83 SPB neurons uniquely form synaptic terminals within this dorsolateral and externolateral subnuclei, in addition to the IL. So these results suggest that these two SPB neurons innervate PVN in a zonally segregated manner. So to test the functional significance of this distinct anatomy of PVN innervation by these two types of projection neurons, I performed a series of gain of function experiments by optogenetically stimulating the synaptic terminals of these two projection neurons within the PVN. So strong activation of axon terminals of either TECR1 or GPR83 neurons produced a robust increase in their locomotion, reminiscent of the escape locomotion in response to the painful stimuli. Interestingly, TECR1 stimulation promoted this backward locomotion, whereas GPR83 stimulation promoted the forward locomotion. In addition, strong activation of axon terminals of TECR1 positive spinal parabrachial neurons also induced this pronounced the jumping behavior. So animal try to jump out of the chamber. And here's a summary plot. And then next, we also examine the pupillary reaction after activation of these pathways. Pupillary reaction is a robust and quantitative readout for the change in autonomic responses. So here I'm giving a 10-second simulation of these pathways. So as you can see here, robust, it automatically increased the pupil dilation, both at TECR1 and GPR83 activation. An interesting difference between these two was that TECR1 activation had this prolonged and sustained effect on the pupil dilation, where GPR83 stimulation had more transient effect in the pupil dilation. And this is a summary plot. So these results together suggest that strong activation of both TECR1 and GPR83 neurons induce the nociceptive behaviors, but differentially influence the mode of escape locomotion and the temporal dynamics of the autonomic responses. Next, we examine their response properties in collaboration with the Sarah Ross Recorder's lab, using this ex vivo skin spinal cord preparation. The Junichi, the postdoc in their labs at the time, performed the wholesale patch clamp recordings on these TECR1 or GPR83 SPB neurons while applying different types of stimuli onto the skin. So he measured the change in the firing rate upon the application of different somatosensory stimuli, including the thermal stimuli here. So in general, TECR1 positive SPB neurons responded, were more responsive to the thermal stimuli compared to the GPR83 neurons, especially the temperature range, the innocuous temperature range, including 40 and 15 degree. Whereas both of these populations really responded well to the noxious range temperatures, including 54 degrees. And here's a summary plot. So these results suggest that both of these populations can convey nociceptive and noxious signals to the brain. To summarize the first half of my talk, so TECR1 and GPR83 SPB neurons form parallel ascending circuit modules. They cooperate to convey nociceptive signals to the brain. And I showed you that they are largely non-overlapping populations, and they innervate the PVN in a zonally segregated manner and produce distinct escape locomotion and autonomic responses. And also, they both respond to the noxious cutaneous stimuli. And for the second part of the talk, I'll focus on this novel dedicated touch pathway in the anterolateral tract. So a textbook view of the ascending touch pathway emphasizes this dorsal column medial lemniscal pathway, where touch signals are first detected by mechanosensory neurons in the periphery, and these touch signals are transmitted to the brainstem through this dorsal column pathway, and then transmitted to thalamus and somatosensory cortex. And this is the traditional touch pathway. In contrast, prior studies on the entorolateral pathway have been mostly focused on the pain and temperature aspect. However, we think that this newly identified GPR83 SPB, the spinal parabrachial pathway, may be the dedicated touch pathway within the entorolateral tract that convey tactile information from the spinal cord to the lateral parabrachial nucleus. And in turn, PBN broadcasts this touch signal to other regions in the brain, including amygdala and hypothalamus. So I'm going to walk through some of the data that support this idea. So going back to our ex vivo recordings, so one of the most unique response properties of the GPR83 projection neurons was that they are highly sensitive to mechanical stimuli. So here in this summary rate plot, the left panel is percentage responders. The right panel shows you the firing rate change upon the application of different somatosensory stimuli, including the temperature and the mechanical forces using the bone fry filament skin indentation. So as you can see here, most of the GPR83 positive projection neurons responded well to a wide range of mechanical stimuli. And when they respond to the mechanical stimuli, they responded with a much higher firing rate compared to the TEC-R1 projection neurons. So these results suggest that really the GPR83 projection neurons are really highly sensitive to mechanical stimuli. And this is a summary plot. So to define the spinal cord circuit mechanism underlying this mechanosensitive response properties of GPR83 neurons, I examine the primary sensory neuron input onto these projection neurons, particularly focusing on the mechanosensory neuron input onto these neurons. So for this, I performed the channel-rhodopsin assisted circuit mapping experiments with acute spinal cord slice preparation using this rather complicated quadruple transgenic mouse lines in combination with the retrovirus injection, which enabled us to specifically stimulate different subtypes of primary sensory neurons while recording from specific subtype of projection neurons. So axonal terminal stimulation of mechanosensory neurons, including MIGB4 positive non-peptidergic neurons, and the TRAC-B positive A delta low threshold mechanoreceptor neurons evoked large analytic recurrence and also action potential firing in GPR83 neurons, but not in the TEC-R1 positive SPB neurons. So these synaptic input analysis suggested the GPR83 neurons receive strong synaptic inputs from the mechanosensory neurons. And this can explain its unique mechanosensory sensitive response properties. And then we turn to the anatomy of this spinal parabrachial pathway. What is really interesting about this spinal parabrachial pathway is that they have unique organizational properties distinct from other ascending pathways in the entorolateral tract. So here in this experiment, I injected cre-dependent alkaline phosphatase virus into the unilaterally into the one side of the spinal cord, and then performed the tissue clearing and the Holman AP staining to visualize the axonal projection going up to the brain. So as you can see here, the PBN receives predominantly bilateral inputs from the spinal cord, whereas other brain targets such as superior colliculus or PAG in the midbrain receives predominantly contralateral inputs from the spinal cord. And another unique anatomical feature of the SPB pathway was that they lack the somatotopy organization. So in this experiment, I injected AAV viruses expressing symptophysine tagged with the 3 different fluorophore into cervical, thoracic, and lumbar spinal cord, and examined their terminal distribution within the PBN. And interestingly, these 3 different colors of terminal originating from 3 different axial levels of spinal cord, they're all intermingled within the PVN, and suggesting that there's no clear somatotopic organization in the spinal parabrachial pathway. So these unique anatomical features led us to suspect that maybe the spinal parabrachial pathway convey affective component of somatosensation rather than discriminative component of somatosensation. And interestingly, in the human patients with chronic intractable pain, such as terminal cancer pain, the patients often receive this anterolateral transection surgeries to reduce their pain sensation. And oftentimes, these patients lose the pleasurable feelings of gentle touch in addition to the pain sensation, while maintaining their ability to locate and identify the nature of the touch signals. So these human studies suggest that maybe anterolateral pathway convey the gentle touch signals associated with the positive valence, as well as pain signals associated with the negative valence. To directly test this idea, I performed optogenetics coupled instrumental conditioning assay, where animals receive optogenetic stimulation of synaptic terminals of this SPB pathway when they press active lever, but not inactive lever. So when I say affective component of somatosensation, it is a bit vague and abstract, but because we cannot ask animals how they feel, so really what we wanted to test here was whether animals like or dislike the activation of these pathways. So here, so I coupled the level process with optogenetic stimulation with different strengths of optical intensity. So, and I started at the high power stimulation and then kind of dialed down the optical strength. And down to the 1 milliwatt stimulation of either GPR83 and TECR1 SPB pathways reduced the level process over time. So the x-axis is multiple day sessions, suggesting that strong activation of these pathways serve as punishment signals, so which is aversive. However, when I further down reduced the intensity of the optical stimulation to 0.4 milliwatt, this weak stimulation of especially GPR83 pathway promoted positive reinforcements. In other words, increased level process. And this increased level process maintained several days after the light on session. So these results suggest that GPR83 SPB pathway can be associated with positive or negative valence depending on the stimulation intensity. To summarize the second part of the talk, this newly identified GPR83 SPB pathway constitutes the mechanosensory limb of the anterolateral pathway that conveys an affective component of touch. So I showed you that these neurons are highly sensitive to mechanical stimuli, and they receive strong synaptic inputs from the mechanosensory neurons. They form bilateral and non-somatotopically organized synaptic inputs to the PVN, and they induce the behaviors associated with either positive or negative valences in a stimulus intensity dependent manner. So the grand summary overall, our study revealed these two anatomically and physiologically and functionally distinct spinal parabrachial pathway that underlie touch, affective aspects of touch and pain. So with that, I'd like to thank first David for his awesome mentorship. I learned a lot from him, and all the people from Ginte Lab that contributed to the work, and our collaborators, especially Sarah Ross and Rick Corbo's lab and Junichi. They did ex vivo physiology for us, and mouse science, and a virus and the funding sources. And with that, I'll take any questions.
Thank you, Sebastian. Oh, go ahead, David.
Yeah, I'll go ahead here for a second. That was a really awesome talk. Thanks for that. So maybe I'm just going to kick it off here to get the ball rolling. And I wanted to just, one of the really cool things that you showed here was that this projection from both of these populations of neurons from the spinal cord to the peribrachial nucleus, but they lacked both a somatotopic organization and they projected bilaterally. And you use that to motivate their potential significance for affective touch. And I guess I have a couple of questions. One is, I think if I remember correctly, you showed that both of these neuronal populations lacked those. The
I was bilaterally. A little bit disconnected. Can you repeat the
Oh, sorry. I was just talking to myself. Anyway, yeah. So I think you showed that both of these, the DPR83 and the TACR1 neurons, both had a bilateral projection, and they lacked somatotopy, suggesting their role in affective touch. Yet when you did the optogenetic experiment, you found that you had these diverging results where that suggested that maybe only one of them was involved with affective touch. And I'm wondering if you could comment a little bit on that, but also maybe comment too on, like, aside from affective touch, what do these projection patterns, what other aspects of touch would you predict might be conveyed through a projection that lacks somatotopy and lacks laterality?
Yeah, so that's a good question. So I think in general, people think that in terms of the different components or aspects, when people think about the touch perception, they are mainly divided into, I guess, the discriminative component and the affective component. So discrimination, I think, that component is conveyed to the brain through the traditional pathway that I described. One of the slides, the physical medial meningeal pathway that goes all the way up to the centric cortex. And I think this spinal parabrachial pathway is uniquely poised to convey other aspects, which is, I think, the affective component. So basically, make animals feel good or bad about given touch or other semi-sensory stimuli. So the reason, so I showed experimentally that is the case in terms of whether or not the animals like or dislike this stimulation of these pathways. But if you look at the anatomy of this parabrachial nucleus, I showed you that these two pathways innervate kind of segregated manner. And some of the main brain targets of this PVN is amygdala and hypothalamus. So I think that connection to the limbic system kind of makes sense. These pathways connect to the limbic system and more involved in decoding the affective aspect of the touch signals. Yeah, so that is
cool. Thanks. Okay, so I might hand it over to Jenny now, who is gonna, I think, field some questions from the audience.
Okay. So first, great talk. I'm gonna go ahead and start off with the first question here that I also actually really would like to know the answer to, which is from Raphael. He says, great presentation. What are the possible mechanisms allowing this dual response of the neuronal population? And I think, like, before you go on to answer that, I just want to say, like, yeah, a little bit more about that, where, you know, you looked a lot at the anatomical projections and some differences with the behavior, but given that you're using the same kind of cell intrinsic channelrhodopsin stimulation of those cells, it seems like this valence switch is further downstream of these cells themselves. And so I think it would be really, I mean, maybe you have some ideas for how this could happen.
Yeah, that is really a good question. So actually, I didn't have time to show the data here today. But one thing that we did was by varying the intensity of the stimulation and look at the fast activated neurons within the PVN. So one thing that I observed with the different strength of the epigenetic stimulation was that the neurons in one of the subnuclei, the external lateral subnuclei, which is one of the main targeted with the GPI83 projection pathway, the neurons in there seem to be depending on the stimulation intensity, the number of neurons activated were changed, and then there was a correlation between the number of neurons activated and the stimulation intensity. But I think what is happening there is that so probably within this PL of the PVN, there are different heterogeneous population of neurons. Some of them are low threshold neurons, some of them high threshold neurons. They have different sensitivity to this like stimulation. So basically increasing the stimulation intensity, you recruit more number of neurons engaged. And then depending on the number of neurons they're activated, that could potentially activate distinct downstream pathways. Actually, this change in graded variable, which is the number of neurons activated, into the transformed into the discrete action selection phenomena were observed in other brain circuits, including the hypothalamic. In context of the depression behavior, there was some reports from David Anderson reporting the dramatic switch in their behaviors, and that depending on the scalability of the number of neurons. So I think kind of similar thing is happening in the PVN here too. So, but one thing that really we wanted to do in the future was actually giving the natural stimuli and then recording or looking at the casting activity of different neurons in the PVN and how their activities are correlated with the natural, pleasurable touch or the painful touch to neurons. So that is an interesting future direction to go.
Cool. And I think to keep us on schedule, correct me if I'm wrong, I'm just going to take one more question. I'll take the next upvoted one here from Erica. She asks, it seems you looked specifically at cells relaying bodily nociception, but not facial nociception. Did you also look at expression of TACR1 and GPR83 in the trigeminal nucleus or trigeminal ganglion?
Oh yeah, that is a good question. So my this study mostly focused on the spinal cord neurons, but when I checked their expression in the trigeminal nucleus, they represent this the facial the stimuli getting onto the facial regions. They are also expressed in the trigeminal nucleus. So I think kind of similar. The circulation is working in the trigeminal system as well, but that is something that we can directly test in the future.
Cool, thank you.
All right, thank you, Sebastian.
Okay, yeah.
Let's see.
Sebastian, we're gonna get rid of your screen and invite Silvana up.
Hi, everyone.
Cool. Great. Okay, Jonah, I'm gonna go.
Hey, Silvana.
Okay.
After a series of glitches, I think I finally fixed my presentation. Let me see if I can share my screen.
Yeah, great.
Amazing.
We see all of your slides, though.
Oh, okay. Let me see. So if I do play from start, you still see all my slides?
We still see all of your slides.
So,
all right.
Just Sebastian had this this problem. If you do presenter view, if you play it like in presentation view, right, and then I think share screen, I think then it works. So try doing that before you share screen.
All right.
Yeah, I hope that works.
Let me see that.
I don't know much about Crowdcast, but
okay. So play from start, and how about now?
Hey, there you go. Cool.
Amazing.
So yeah, your echo seems to be gone. So, okay, I'm going to take an opportunity here to introduce Silvana. So it's really my honor to introduce Silvana. She is here at my same institution, NYU. Silvana's a postdoc in Rob Fromke's lab at the Skirball Institute at the New York University School of Medicine, where she studies how the brain changes during parenting using the mouse as a model. Silvana earned her bachelor's, master's, and PhD at Pierre and Marie Curie University in Paris. When she was there, she worked on mechanisms of synaptic plasticity in the striatum. Since moving to NYU in 2017, she has, like Sebastian, has received several awards, including a Leon Levy postdoctoral fellowship. Most recently, she received a NARSAD Young Investigator Award. And in addition to her really exceptional science, Silvana is extremely engaged in the neuroscience community. She's organized meetings, including the Cross-Institutional Sense to Synapse meeting, which is here in the New York tri-state area. She's a mentor to young scientists, and she's volunteered her time to help educate the public about science in the brain. So in short, Silvana really is a force of nature, and I'm looking forward to hearing her talk about plasticity in hypothalamic circuits for oxytocin release.
Thank you so much, David. And thanks for giving me the opportunity to speak today. I'm sure we're going to have a lot of fun during this first season. I'm going to present you my work for the past 3 years or so in the Fromke lab, where I work on hypothalamus and maternal behavior. We all know that mammalian infants are very needy, and they emit a variety of sensory signals in order to attract caregivers. So the job of new mothers is to very rapidly learn how to detect and interpret those signals in order to engage in appropriate caregiving responses. So different sensory stimuli from infants will elicit different types of maternal behavior. But for the rest of the talk, we're going to focus on behaviors that are modulated by the neurohormone oxytocin. Oxytocin has been described classically as a nursing hormone. And the nursing occurs mainly by a direct somatic touch with the infant. But for the rest of the talk, I'm going to show you some evidence that actually infant vocalizations can also trigger oxytocin release in the maternal brain. But what is oxytocin anyway? Oxytocin, as I mentioned, is a neurohormone synthesized and released from the hypothalamus. And here is a section of the mouse brain showing one of the nuclei synthesizing and releasing oxytocin. This is the paraventricular nucleus, or the PVN. There is another one which is involved, which is the SON, but we're going to focus for the rest of the talk on the PVN because PVN oxytocin neurons are those sending oxytocinergic projections to a variety of central brain areas. As you can see, there are a lot of sensory, limbic, and motivational areas receiving very dense oxytocinergic projections. Well, the thing is, we have very limited knowledge about when and how oxytocin neurons are activated in order to release oxytocin in downstream areas. And actually, the only circuit that is well described is how oxytocin neurons release oxytocin in the blood. And that comes from the nursing literature. Briefly, what is happening is that oxytocin from the hypothalamus of mothers gets released in the bloodstream in order to facilitate the milk let down. And that happens by somatic stimuli from the nursing infant. And Sebastian just told you a lot about the spinal cord and brainstem circuits that can relay this kind of information. Well, curiously, in this case, milk letdown can happen in the absence of the nursing infant. And this is something that has been very well described in humans. So human mothers report that they can experience spontaneous milk letdown only by hearing the sound of a baby cry. And there are human studies showing that baby cries can activate the hypothalamus. I know you're thinking it's a torture to be in the scan listening to baby cries, but what is important for us is that this activation is strong enough to also trigger oxytocin release in the blood. So that tells us that infant vocalizations are extremely powerful stimulus to activate nursing circuits, but we have no understanding about how that's happening. And actually, the only thing that we know about how vocalizations are processed in the maternal brain comes from studies in the auditory cortex, mainly. And here I'm going to show you what we are doing in our lab. We study maternal behavior in mice. And so mice are very curious because they exhibit this very stereotypic pattern of maternal behavior known as retrieval behavior. Briefly, what is happening is that when a mouse pup is isolated from the nest, it will cry, it will emit these ultrasound distress calls. You can see the spectrogram here. They will attract the mom, and the mom will fetch the pup and bring it back to the nest to safety. And very importantly, the neural responses at the cortical levels are extremely robust, such that if you present temporary morphed pup calls, they're preserved. So here I'm just showing you an example of a cortical neuron that I recorded in the primary auditory cortex. And you can see these patterns of synaptic excitation and inhibition to standard, but also temporarily morphed calls are preserved. So the processing of these vocalizations is extremely robust. The thing is, we have no idea how auditory stimuli can actually reach the hypothalamus and activate hypothalamic neurons in order to release oxytocin. And I'm going to show you that this indeed can happen, and that oxytocin neurons are not just a part of this autonomic circuit to regulate milk let down in lactating females. So I'm going to show you for the rest of the talk that oxytocin neurons can be activated by pup calls, what are the auditory pathways conveying auditory information to the hypothalamus, as well as what are the plasticity mechanisms that support these responses. Oh yeah, and finally I'm going to dig a little bit into what is the implication of these circuits for maternal behavior and oxytocin release. So we started by performing cell-attached and whole cell recordings in the PVN of awake dams. So dams, these are maternal mice. For the rest of the talk, I'm going to refer them to them as dams. Our electrophysiological setup is also equipped with an ultrasound speaker via which we can play these distress vocalizations. The thing is that this technique took a while to develop for several reasons. So first of all, PVN is a structure which is quite deep in the mouse brain and really small. But the real challenge came from the fact that hypothalamus is an extremely heterogeneous structure, and you can find a variety of cell types in there, and you can appreciate here that the oxytocin neurons are just a very small fraction of the total amount of cells. So in order to be able to identify and record from those cells, we adapted a method that actually has been developed at NYU from the Rudy lab called channel rhodopsin assisted patching. It can be pronounced trap or crap. The important is that the crap is very handy because it actually allows us to identify those neurons. So what we do is to place an optical fiber in the patch pipette connected to a blue laser, and then we can patch in animals expressing channel rhodopsin under the oxytocin promoter. So each time we encounter a cell, we're going to shine brief pulses of blue light, and oxytocin neurons are identified because they're going to spike very reliably in response to those pulses. Whereas the rest of the hypothalamic cells will basically remain silent. So this is what we have. We recorded pup calls from isolated pups with ultrasound microphone. We have this library of calls that we loop in a sort of a podcast and we'll play to awake dams while we record from their PVN. What we found is the following. So here I'm showing you cell-attached configuration of one identified oxytocin neuron. I want you to know that those neurons, they fire at very low firing frequencies at baseline. We then present a series of pop calls. In this case, these are 15 pop calls separated by a second, and each call lasts for a second. We then record a second baseline following the playback of pop calls, and we were very surprised actually to find that oxytocin neurons drastically increase their firing rate following the vocalizations. And you can see in this specific example, that cell increases its firing rate of about 4 to 5-fold. And even more interesting for us was the fact that we did not find this effect in other hypothalamic cells. So this is an example from a non-oxytocin neuron, so this is a non-identified PVN neuron, which does not increase its firing rate following the playback of calls. And here you can see the histograms of both neurons. What we see at the population level is the following. The majority of oxytocin neurons increase their firing rate following or even during the pop call presentation at the different time points, but just dragging the mean of all these neurons towards increase in activity. And that's not something that we find in the rest of the hypothalamic cells. So that tells us that there is some sort of a cell type specificity about the processing of vocalizations in the hypothalamus. So then we wanted to find out what are the auditory inputs that signal to oxytocin neurons. We performed cell type specific rabies in oxy-3 mice. So these rabies will jump one synapse backwards and will stay in all areas that project monosynaptically to oxytocin neurons. And we were specifically looking for auditory relays. Very surprisingly for us, classical relays such as the inferior colliculus, auditory cortex, also the medial geniculate did not project to oxytocin neurons. But we found a very dense projections from the posterior infralaminar thalamus. So for the rest of the talk, I'm going to refer to it as the PIL. So PIL has been described as a part of the non-lemniscal auditory pathway. Well, however, all the experiments done in the PIL are only investigating responses to synthetic sounds. So this is white noise or pure tones. So we wanted to know if PIL indeed responds to vocalizations. We found CFO staining in the PIL after dams were exposed to pop calls, as well as when we record a multi-unit activity with tungsten electrodes, we saw that this area increases its firing rate during the calls. And right now I'm doing some cell attach and whole cell recordings, looking more into depth of these PIL auditory responses. The thing is, PIL, it's a very good candidate of being an auditory relay to the hypothalamus. So we wanted to characterize the circuit more into detail, and it was great that Sebastian already gave an overview of channel rhodopsin circuit mapping that we're doing here. So it is the same. Basically, we're injecting channel rhodopsin in an area of interest. In this case here, this is the inferior colliculus. Then we slice the acute brain slices of the target area, the PIL in this case, and we investigate optically evoked responses in order to check if these connections are not only anatomically present, but also functional. So we found that the inferior colliculus projects very reliably to the PIL. We found much less reliable and also kind of sparse and small amplitude connections coming from the auditory cortex. So these are corticofugal projections to the thalamus. Most importantly was that the PIL signals to the PVN, specifically oxytocin neurons, in a very reliable fashion. So this thalamus to hypothalamus pathway seems to not be, only anatomically present, but it's also functional, meaning that the thalamus is able to activate oxytocin neurons. So here's what we have. I showed you we have a way to identify oxytocin neurons in like animals. I showed you that those oxytocin neurons respond to call by this delayed increase in firing, and that they receive projections, auditory projections, supposedly by this non-canonical auditory pathway. Well, now the question is how these responses arise. Again, they're very slow to be due to synaptic integration because they happen in orders of tens of seconds to minutes. They're too slow to be auditory responses per se, meaning that oxytocin neurons do not exhibit spikes that are time-locked with stimulus presentation. So we thought that this should be due to some sort of plasticity, long-term plasticity mechanisms coming from when the thalamus is discharging, something is happening with oxytocin neurons. So what are these mechanisms? We hypothesized that the increased output of oxytocin cells can be due to several things. First, it can be due to increasing their excitability following thalamic discharge during the pop calls, increase in excitation, or decrease in inhibition. And I will spare you all the negative data we had, but briefly what we found was that decreasing inhibition is likely the mechanism that promotes these responses. So actually oxytocin neurons get disinhibited following calls. How we found that? We injected channel rhodopsin in the PIL with cut acute brain slices of the PVN in mice, which express TB2 in oxytocin neurons. So we can identify them, touch from them. We have an electrical stimulation in the slice that helps us track synaptic excitation and inhibition. And on the top of that, we have the auditory PIL fibers tagged with channel rhodopsin. So when we optogenetically stimulate them with a very similar pattern of activation as the one that we see during in vivo when the thalamus is discharging, so we kind of mimic this pattern in slices, we see that synaptic inhibition drastically decreases, although excitation stays the same. And that leads us to a long-term decrease in the inhibitory to excitatory ratio. In our case, we recorded to up to 20 minutes, and this decrease in inhibition was stable. So we think that the disinhibition of oxytocin neurons is actually likely to underlie those increased responses in vivo following calls. I will not get into the details of all the following experiments that we performed, but basically we found that this decrease in inhibition happens because there is a internalization of postsynaptic GABA A receptors onto oxytocin neurons that happens when thalamic fibers are discharging. We also now are looking at how that can happen in vivo. So we know that NMDA receptors require not only presynaptic glutamate, but also postsynaptic depolarization in order to be activated. So right now we're doing wholesale recordings in oxytocin neurons, looking at their depolarization during pop calls, which might be a mechanism to release the magnesium block and activate NMDA receptors. And I want to point out that these depolarizations is not something that we see in the rest of the hypothalamic cells. Well, our next question was what can be the physiological relevance of this whole mechanism? And we started by looking specifically at pop retrieval behavior. As I showed you, this behavior relies on detection of calls in the maternal brain. So we started by blocking the PIL to PVN connection, just inhibiting with inhibitory dreads, and looking at retrieval. So this is the work of Haban Issa, who is a graduate student in the lab. And what Haban found was that in dread animals injected with CNO, they perform at the same rate as control animals, meaning that they would pick up pups at the same amount of times. However, those animals showed increased latency to retrieval. So basically what we're seeing here is the mom wandering around and not being very interested in picking up the crying pups. So they take really long time. So we then hypothesized that the PIL to PVN pathway would be involved in speeding up maternal behavior because it will promote activation of hypothalamic oxytocin neurons, which then in turn will induce oxytocin release in downstream areas. And talking about motivation, we decided to check if indeed pop calls are able to induce oxytocin release in motivational areas such as the VTA. So here we are using a newly developed genetically encoded oxytocin sensor developed by the Kwon lab at Johns Hopkins. And because I will not have time to describe the mechanism of how this works, I invited to check their bioarchive paper for the details of this construct. Basically, it consists of injecting a cocktail of 3 viruses in an area of interest and also implanting an optical fiber because the activation of this sensor depends on blue light. Then after expression, the idea is to expose animals to behavior or some sort of paradigm that is supposed to release oxytocin in this target area. In our case, we expose dams to passive listening of pop calls while we also shine blue light in the VTA. And that will enable the sensor to capture oxytocin release. What we're seeing here, this slice of VTA, all the red cells are those which express the oxytocin receptor, the oxytocin sensor, and all the green cells are the cells that have detected oxytocin release. And right now we're still doing the quantification. Yes.
I'm trying to finish up in the next minute.
Oh yeah, sure. That's my last slide.
Just a heads up.
Thanks.
Cool. Yeah.
Preliminary data, we're still doing the analysis here, but basically we're counting the ratio of green to red to see what amount of cells from those expressing the sensor were able to detect oxytocin release in the VTA via this pop call presentation. And this is my summary slide. So briefly, I showed you that oxytocin neurons can get activated by vocalizations. And this most likely happens because they get disinhibited after thalamic discharge during the pop calls. And this disinhibition happens because GABA receptors on the postsynaptic side get internalized, so inhibitory transmission decreases. We also suggest that this thalamus to hypothalamus pathway and this increase in oxytocin firing can speed up maternal behavior and can also trigger oxytocin release in downstream areas. And by that, I will wrap up. I want to thank my entire lab. We're more than 20 people, so I'm certainly not listing everyone. Rob Fromke, he's an amazing mentor. He gave me green light to develop this new avenue in his lab. A lot of people from NYU also outside and my funding sources, and I'm very happy to open a discussion.
That was great, Silvana, thanks.
Should I stop sharing?
Yeah, I think that's fine. So one of the hallmarks of a really great talk, I had a lot of questions throughout, and I was typing down my questions, being like, "Oh, this is so great! I want to know more." And then the next slide, you'd answer them, which is fantastic. I love it. There's so many things I want to know more about. One of the things that I guess maybe this is a softball question for you, but you did all of these experiments in dams. And I was really blown away by the specificity of the projections from the PIL onto the OXT expressing neurons, oxytocin producing neurons in the PVN. And I'm wondering how you think that connectivity, that uber specific connectivity arises. Is it something that has to do with the experience of being a mother? Have you looked in male mice to see whether you've seen the same connectivity or in virgins, for example?
Yeah, so I think there's two parts of the question, actually. First, why oxytocin neurons get activated but not non-oxytocin neurons. I'm now doing some connectivity studies to see if other cells are connected to the PIL. What I think is happening is that even though they're connected and they exhibit optic currents, those currents do not have NMDA components. So they actually cannot have this long-term triggering of plasticity. In terms of virgins versus dams, I've done a little bit experiments in virgins, and I cannot really claim what's happening there. My sense is that they have very sparse responses to calls, so they cannot really sustain this increase in firing. Why that happens? That can be several things. So first, there are a lot of plasticity happening during motherhood in the PIL itself. So that can be inherited upstream. That's one possibility. The other possibility is that what's happening in dams, actually in the hypothalamus, you have this very prominent retraction of astrocytic end feet that favor intersynaptic crosstalk and glutamate spillover. And those NMDA receptors are supposedly located at extrasynaptic sites. So I'm actually doing experiments right now. So this stripping of the synapse from the glia in dams can actually promote the integration of those PIL inputs by the NMDA receptors. In males, I have no idea. That's something I have to do. But we hypothesize that this whole circuit is something generalized, but the strength of those connections can vary depending on the experience of the animal.
That's great, thanks. Okay, I'm gonna hand it over to Jenny, and she's gonna deal with the questions that we had from the audience.
Yeah, awesome. So I think you've addressed some parts of some people's questions in the chat here. I think one additional idea or theme that people seem to have is like, how specific is this kind of response to pop calls? So for example, are these oxytocin cells sensitive to white noise bursts or other vocalizations that are not pop calls? And conversely, do you find a consistent decrease in the non-oxytocin cells during pop call?
Okay,
presentation.
I'm gonna break out the question. I've done experiments presenting pure tones, not white noise though. What I see is that oxytocin cells, they do not have, preferred frequency as what we see in auditory areas. They also do not increase their firing rate long-term, actually not at all, after a sequence of pure tones. Why that can happen? Two possibilities. One, it happens actually in the PIL, and the PIL is kind of heterogeneous, so you have cells that are responding to calls but not to tones, and vice versa. I can imagine that the wiring is such that the PIL neurons responding to calls preferentially project to the thalamus, but those responding to pure tones are actually projecting to the amygdala. There's a lot of research on these connections. That's one hypothesis. The other one is that the discharge of PIL during pure tones does not, does the same pattern of activity as the discharge during calls at a synaptic level. So actually, it's not strong enough to trigger this NMDA activation. Yeah. Concerning other calls, I've done exactly 2 experiments, doing like 2 cells, you can imagine, so it's very anecdotal, presenting other types of calls, which are wriggling calls. So those are calls which pups emit while they're hungry. So they're not in distress, they're just hungry. And I see similar activation of oxytocin cells long-term. So that's very curious. I don't know about adult vocalizations though, but I can imagine. Yeah.
Cool. I think we're running at just about an hour now. I think we still have a ton of really interesting questions in the chat. Jonah, what do you think?
I don't know what the rules are, but I think you should go ahead and ask a couple more questions. Maybe take another couple of minutes.
Okay, cool.
2 or 3 minutes. I don't know. Go for it.
Yeah. Okay, so part of this job, I guess, is I see a lot of upvotes on this first question, but I feel like this is something that was somewhat addressed. If the person who asked this wants to insist on it being answered, just let me know. But I'm going to move on and ask some of the lower questions here. So Joanna says, nice talk. Do you think that PVN VTA pathway is what drives the motivation for pups retrieval? Also, does it drive oxytocin secretion to the blood as well?
So VTA is certainly involved in maternal motivation and pup retrieval. The thing is, it's not the whole story, right? It's a very distributed network, so it's really difficult to block pup retrieval by just inhibiting separate areas, which makes sense because evolutionary, it's important that we have a lot of redundant circuits that lead to the same behavior because for the survival of the pups, that's extremely important. We picked VTA for actually several reasons. The oxytocinergic projections are very dense, and also VTA is kind of easy to target. For example, it's easier than SNC, right? So for us, that was just a good readout. I do not know if the same neurons could release, for example, in the amygdala, lateral septum, or elsewhere. About the blood, I would love to know that. It's actually very difficult to target by patch specific populations of oxytocin neurons because they're already super sparse. They're 10% of PVN neurons or less. But by fiber photometry, we can address that. So there is a way to specifically infect with GCAM oxytocin neurons which project to the bloodstream. And I'm actually currently doing some photometry experiments. I cannot tell anything because we're still struggling with the GCAMP expression though. But yeah, that's something to look at.
I see. That sounds super cool. Okay, one more question. Okay, I'm gonna take this one from Lena and combine it with something that I also had a question about. Lena says, very impressive. Have you tried recording the inhibitory response in vivo during pup calls to determine the time course of its reduction? And she also asks, where is this inhibition coming from? And my related question is that you showed kind of in your background slides that the peak of oxytocin release is actually prior to the beginning of suckling in, I think, like non-human primates. And it seems like this is a very like unidirectional, like non-reversible kind of plasticity, at least what you see in slice up to like 20 minutes later. And so what is the mechanism for eventually like terminating or decreasing the response of these oxytocin neurons, or this change in plasticity, let's say?
About the inhibitory currents, I've tried to do voltage clamp recordings in the PVN. It's a nightmare. I've done some on non-identified cells though. But I cannot say anything about oxytocin cells. The thing is that they're so difficult to find that I even struggle with the whole cell recordings of spiking responses. So I do not know. I would love to do that. One thing about the source of inhibition, that's something that intrigues me for a very long time. So the main source of inhibition is actually coming from the lateral hypothalamus. But there are also long-range projections from amygdala and septum. So right now what I'm doing is that I'm trying to inject crimson instead of channel rhodopsin in one of those areas and having channel rhodopsin in the thalamus and actually tracking which source will decrease. Yeah, doing these double wavelength experiments. And I would love to know. I guess it's the lateral septum, but I don't know. About the long-term effect, so you can imagine on the slice, even though it's alive, it's kind of silent. So in vivo, there are a lot of things happening. I still don't have an understanding what would be the homeostatic mechanism that will bring those responses back down. I can imagine several things. For example, these lateral hypothalamic cells, which are GABAergic, they might have some control, and they actually even we can imagine that they go suppressed during calls, but they rebound, and they actually suppress oxytocin activation later. But that's all just sorts of stuff that I imagine. Or amygdala and septum, they're very, very strong inhibitors of oxytocin neurons. So yeah, things happen in vivo that we cannot see in slices, certainly.
Yeah, I get that. Thank you so much.
Okay, well, I think we have to end it here, but I just want to remind everyone that hopefully, you come to the next sessions and we keep growing this into a bigger and bigger stable program. I thought these talks were amazing. Silvana, that was a great talk, and Sebastian too. Those were like just really interesting neuroscience that I just don't know anything about. So with that, I'd like to end the session, and thanks everyone for coming, and we'll see you next time. You can look at our website for the next scheduled session, and there will be a bunch more. We'll renew the season after that, hopefully. Thanks to everyone for coming.
Thanks for organizing.
I don't know how to, okay, and broadcast.